Dear students, our lesson for today is about mitosis. Before we start, please subscribe and share this channel with your friends so we can all learn together. Have you ever wondered how you grew from a tiny baby into a student, or how your skin heals after you scrape your knee? The answer is mitosis. Mitosis occurs in somatic cells, which are also known as body cells. This includes almost every cell in your body, from your skin and muscles to your bones and internal organs. In these cells, mitosis is the process where one cell divides to create two identical copies of itself. This is how we grow, replace old cells, and repair damaged tissue. To understand mitosis, we first need to look at DNA. Our genetic instructions are organized into structures called chromosomes, located inside the cell's nucleus. In a normal human body cell, we have 46 chromosomes. We call this a diploid cell, or 2n equals 46, because we have two sets of 23 chromosomes, one set from each biological parent. Before a cell can divide, it must make a copy of everything it contains so that both new cells have the full set of instructions. This happens in a stage called interphase. Before the division actually starts, the cell must prepare. This happens in a stage called interphase. It is important to remember that interphase is not part of mitosis or the actual division. It is simply a preparation phase where the cell grows and performs DNA replication. In fact, interphase takes much more time than mitosis itself. The cell spends about 90% of its life in interphase just getting ready. During this time, the cell performs DNA replication. Each chromosome is duplicated, resulting in two identical molecules called sister chromatids, which are attached at a point called the centromere. Once the cell has finished its long preparation, it is ready for the four main stages of mitosis: Prophase, Metaphase, Anaphase, and Telophase. You can remember them with the acronym PMAT. First is Prophase. During this stage, the chromosomes condense and become visible under a microscope. The nuclear envelope, which acts like a protective shell around the DNA, starts to break down, and the cell begins to build a spindle to help move the chromosomes around. Next is metaphase. Think of M for middle. The chromosomes line up in a single row right along the center of the cell. Each chromosome attaches to the spindle fibers. Then comes anaphase. Think of A for away. The sister chromatids are pulled apart and move away from each other toward opposite sides of the cell. Now each side has an exact, identical set of genetic information. Finally, we reach telophase. Remember T for 2. In this stage, the chromosomes reach the opposite ends of the cell, and the cell prepares to become 2. Two new nuclear envelopes form around each set of DNA, creating two identical nuclei within the single cell. To finish the process, the cell undergoes cytokinesis. But what does this word actually mean? If we break it down, cyto means cell, and kinesis means movement or cutting. So cytokinesis literally means cell cutting. During this stage, the cytoplasm, the fluid inside the cell, pinches in the middle until the cell physically splits into two separate, identical daughter cells. The result of mitosis is simple but amazing. We started with one somatic parent cell and ended with two identical daughter cells. Each one is a perfect clone of the original, ready to help your body grow and stay healthy. I hope this video helps you understand the vital role mitosis plays in your life. In our next lesson, we will look at a different type of division called meiosis. Meiosis is all about variety. It's the reason why every person on Earth is unique. Thank you for watching, and I'll see you in the next lesson to explore the world of genetic diversity.
Like, share, and hit the bell so you never miss a BioBeats Tune video.